So today we're diving into a really cool discovery about where brand new genes come from. And the answer, it turns out, is hiding in our DNA's daily schedule.
All right, so we all know our DNA is constantly evolving, right? But think about this for a second. For evolution to create entirely new things, it needs brand new parts, new genes. Just tweaking the old ones won't cut it. So that brings us to the big mystery. Where do these new genes actually come from? Okay, so to figure this out, scientists basically played detective. They followed two major clues. And the first one, it all starts with a simple but really powerful mistake our cells make all the time. Think of it like a genomic copy-paste error.
And that error has a name: gene duplication. It's exactly what it sounds like. When our DNA is copying itself, sometimes a gene gets copied twice by mistake. Now this is huge because you've got the original super important gene doing its job, and now you have this spare copy. And that spare copy, well, that's evolution's new playground. It can mess with it, change it, and try to create something new, all without breaking the original machine. This is a huge, huge source of new genetic material.
So if this is happening, can we see it? Absolutely. When you look across the human population, you see the results of these recent copy-paste jobs everywhere. We call them copy number variations, or CNVs for short. It just means there are chunks of DNA where, you might have one copy of a gene, but I might have two, or maybe even three. These CNVs are basically hotspots for recent, fast-paced evolution.
And here's where it gets really interesting. The researchers made a crucial connection. They looked at genes we know have been duplicated over evolutionary history, and guess where they found them? Yep, right in those CNV regions. In fact, they're over twice as likely to be in a CNV region as a non-duplicated gene. So this is our first big takeaway. These CNV regions are like the nurseries where new genes are born.
All right, that's clue number one. Now for the second clue, and this one adds a whole new layer: time. But I'm not talking about millions of years of evolution. I'm talking about the few hours it takes for your DNA to copy itself right inside a single cell.
Now you'd probably think that when a cell needs to divide, it just, hits copy on the whole genome and gets it done all at once. But nope, that's not how it works at all. It's actually on a super strict schedule, like a replication clock. Some neighborhoods of your DNA get copied right away, nice and early. And other parts, they have to wait till the very end of the line.
And what's fascinating is that these early and late neighborhoods are totally different. The early replicating spots, think of them as the bustling downtown core. They're packed with important genes, they're super active, and the copying process there is really careful, very few errors. But the late replicating zones, they're more like the wild, undeveloped outskirts. The DNA is packed super tight, there aren't as many essential genes, and the whole copying process is, well, it's under more stress, it's sloppier, you get way more errors and mutations out there.
Okay, so let's recap. Clue number one, new genes pop up from duplications inside those CNV hotspots. And clue number two, our DNA copies itself on a strict schedule, from early to late. Now this is where the big aha moment happens, because when the researchers put these two clues together, they found something amazing, something nobody expected.
They found a powerful, undeniable connection. Those CNV regions, the nurseries for new genes we just talked about, they almost always replicate late, way late in the schedule. So the birthplace for brand new genes isn't in the nice, safe downtown core. It's out in those sloppy, error-prone suburbs of the genome.
And we're not talking about a small trend here. The numbers are just staggering. Get this. If you look at genes that are evolutionarily brand new, like ones that have appeared since our ancestors split from other primates, a whopping 61% of them are sitting in these late replicating zones. That's not just a little bit, that is the dominant pattern.
Yeah, even the researchers themselves said the finding was striking. This wasn't some fluke or a lucky guess. The data was crystal clear. New, young genes are overwhelmingly found in the parts of our DNA that get copied last.
And this chart just lays it all out perfectly. You've got gene age on one axis, replication time on the other. And look at that, the relationship is so obvious. The younger a gene is, the more recently it evolved, the more likely it is to be in that late replicating bucket. And the oldest, most fundamental genes, they're almost always in the early replicating group. It's a direct link between how new a gene is and when it gets copied.
Now you might be thinking, okay, but maybe this is just a weird human thing. Well, the scientists thought of that, so they looked at the mouse genome. And guess what? They found the exact same pattern, the same surprising connection. That tells us this isn't some random fluke. It's a deep, fundamental rule for how new genes are made in mammals.
Okay, so this all leads us to the really big question, why? Why on earth would evolution choose the messiest, most error-prone part of the genome to be its R&D department? It seems totally backwards, right? It should be the safest place, not the sloppiest. Well, let's break down the model the paper suggests, because it turns out that what looks like a bug is actually a feature, a brilliant one. Evolution isn't just being careless, it's being incredibly clever. It's using this area as its own dedicated, messy workshop.
So here's how they think it works, step by step. First, that replication stress in the late replicating zones causes more of those copy-paste errors, creating the new gene duplicates. That's our raw material. Second, these new copies appear in neighborhoods that don't have many important genes, so there's less risk of them messing up something critical. Third, the DNA there is packed so tightly that the new gene is often kept off or silent, so it can't cause trouble right away. And finally, that higher error rate we talked about, it actually helps the new gene mutate and evolve faster, giving it a better shot at finding a useful new job.
So when you put it all together, the big picture is just perfect. These late replicating regions aren't a mistake, they are evolution's workshop. It's this brilliant quarantined sandbox where nature can safely experiment with new parts. It can tinker, it can fail, it can try again, all without any risk of breaking the essential, finely tuned machinery of the cell that's running in those pristine, early replicating areas.
I mean, think about that. It's this mind-blowing link between something that happens over just a few hours inside one of your cells, the simple act of copying DNA, and this grand process of evolution that unfolds over millions of years. It really makes you wonder, doesn't it? What other huge evolutionary secrets are hiding in plain sight, right there in our DNA's daily routine?